Backtracking news, yeah, a technique can determine for the first time how frequently and exactly where a molecular event called backtracking occurs throughout the genetic materials that called genome of any species, a new study shows. Published online February 9 in Molecular Cell, the study results support the theory that backtracking represents a widespread form of gene regulation which influences thousands of human genes, including many involved in basic life processes like cell division and development in the womb. Led by researchers from NYU Grossman School of Medicine, the work revolves around genes, the stretches of DNA molecular letters arranged in a certain order that means sequence to encode the blueprints for most organisms. In both humans and bacteria, the first step in a gene's expression, transcription, proceeds as a protein machine called RNA polymers to takes down the DNA chain, reading genetic instructions in one direction. In 1997, Evgeny Nutlar, PhD, and colleagues published a paper that showed RNA polymers can sometimes slip backward along the chain it is reading, a phenomenon they named backtracking. Studies since then have shown that backtracking occasionally takes place in living cells soon after RNA polymers begins RNA synthesis or when it encounters damaged DNA to make room for incoming repair enzyme. Subsequent work suggested that the backsiding and repair machinery had to work quickly and disappeared, or it might collide with DNA polymers to cause cell death, including breaks in DNA chain. Now, a new study led by Nutlar's team at NYU Langone Health reveals that there are new techniques. Long-range cleavage sequencing can directly detect where backtracking events begin and end by complementing past approaches that were indirect or limited. The new methods reveals that many such events move backwards further than once thought and in doing so last longer. The results also suggest that persistence backtracking occurs frequently throughout genomes, happens more often near certain gene types, and has functions well beyond DNA repair. The surprising stability of backtracking at longer digesters and distances makes it likely that it represents a ubiquitous form of genetic regulation in species from bacteria to human, says Nutlar, the study's senior author and the Julie Wilson Anderson professor in the Department of Biochemistry and Molecular Pharmacology at the NYU Langone. In further work expands our findings to different developmental programs and pathological conditions. Backtracking make akin to epigenetics, the discovery of which revealed a surprising new layer of gene regulation without changing the DNA code. RNA polymers to translates DNA code into a related material called RNA, which then directs the building of the proteins. To do so, the complex moves down DNA chains in one direction but backtracks in certain scenarios. First studies have shown that as RNA polymerase II backtracks, it forces out, extrudes from its interior channel tip the RNA chain it has been building based on RNA and DNA code. As prolonged backtracking is prone to causing detrimental collisions, transcription is thought to be quickly restored by the transcription factor that is called IIS, TFIIS, which promotes the cutting of cleavages of the extruded backtracked RNA. The clears and thus curves the way for RNA polymers to resume its forward code reading. Other earlier studies, however, had shown that when polymers backtracks beyond a certain distance, the backtracked RNA can attach to channel through which it is extruded, holding it in place longer. Locked backtrack complexes are less likely to be rescued by TFIIS driven cleavages and more likely to delay transcription of the gene involved. This led the theory of backtracking, in addition to playing a key role in DNA repair pathways, may dial up or down the action of genes as a major regulatory mechanism. According to the researchers, TFIIS likely occurs at low concentration in living cells and competes with hundreds of other proteins to get at the cut out backtracked RNA so transcription can continue. In the current study, the team instead used a high concentration of purified TFIIS to precisely cut out any pieces of backtracked RNA anywhere it occurs in a cell's genetic code. This made the cutout snippets available to technologies that read code sequence and provide clues to their locations and functions. The research team also found that the genes that control histones, protein pools the DNA chains wrap around within the chromatin that organizes gene expression are highly prone to persistence backtracking. Beyond the report,